Welcome back, everyone. We hope you enjoyed a quick stretch break. We are very excited to host our Richard B. Coletti Clinical Plenary. Our speaker today is Charlie Lees, and the title is Diet the Cause of IBD, and Where Are We with Therapy? I'm going to turn it over to Dr. Sabina Ali for introductions.
Thank you, Julie. Hello, everyone. My name is Sabina Ali. I'm representing the clinical committee with my co-chair, Dr. Brendan Boyle. We are very excited for our next speaker. Dr. Charlie Lees is a professor of gastroenterology at University of Edinburgh in UK. He's been involved in ECHO, in CSO Scottish Specialty Lead for Gastroenterology, and sits on the British Society of GI IBD Research Committee. He is the chief investigator of PREDICT. PREDICT aims to discover the cause of flares in Crohn's and UC in the process of identifying prognostic factors and modifiable environmental and microbiome elements that can target in future interventional study. He's an educator and a global digital opinion leader. You can follow his newsletter called Atomic. You can find him on the YouTube channel, and you will definitely find him on Twitter. Welcome to Improve Care Now, Dr. Lees.
Thanks very much, Sabina, and Julie, and to you all for joining me for the next wee while. So my name's Charlie Lees, as you heard, and I have a number of interests, but core to it is seeing if we can improve the outcomes for people living with IBD today across the world. And today I'm going to talk to you about, is diet the cause of IBD, and how far off is dietary therapy for inflammatory bowel disease? Here are my disclosures. So what I'm going to talk about today is to set the scene a bit about who gets IBD and why, and think about our changing environmental landscape. I want to talk about specific dietary components that might be causative in IBD. And we're going to look at high-fat diet. We're going to look at emulsifiers, ultra-processed foods, sugars, fibers, yeasts, and even herbicides. And then we'll move to think about dietary strategies to prevent IBD in individuals, at-risk individuals, and then think a little bit about diet as a treatment for IBD and what we can practically do today, mostly around advice and the challenges as well as the opportunities for patients, touching on things like adherence. So as a gastroenterologist, the gut is centered to many illnesses, and within the gut, we have this fabulous structure where we have these billions of bacteria separated from this very active immune system by this single layer of epithelial cells. And it's within this context that inflammatory bowel disease develops, when this homeostatic mechanism breaks down. Across Scotland, we're approaching 1% of our population with IBD, as I'll show you. We understand, I think, more and more that this chronic bowel inflammation is a mucosal immune dysregulation with this dysbiotic or altered gut microbiome and these environmental as well as genetic drivers. Over time, the diseases relapse and remit, and do so in an unpredictable manner, not forgetting that approximately 1/3 of people living with IBD will have a really quite quiescent or mild disease course over time. But unfortunately for many, most in fact, there is a need for at least medical therapy, for many the need for surgical therapy. And within this context, we have not just an urgent unmet need, but also many unanswered questions. Starting with who gets IBD and why. And the answer is that IBD can affect anyone from any age, through childhood to the elderly, of any geography in the world, of any ethnicity or wealth or whatever. It doesn't really discriminate at all, inflammatory bowel disease. Globally, we've seen a shift in the environment from the first descriptions of Crohn's disease in the early 20th century, from then what we've seen across the world with this growing incidence and subsequently prevalence of Crohn's disease and ulcerative colitis. First half of the 20th century, not very much, but it was in Northern Europe and North America. This shifts again, And you can see that we start to see more and more IBD in the Far East, the South American and Indian subcontinents. And the future is this as a truly global consideration. Gil Kaplan has given us these 4 stages in the global evolution of IBD. And in the West, in North America, in Europe, including Scotland, we're here in this compounding prevalence phase where incidence is static, i.e. the number of new cases per year is not increasing anymore, but the prevalence, the number of people living with the disease is going up. So here in Scotland, we've looked at this. We found 5 years ago that 1 in 125 people were living with IBD, and we've plotted this out using some modeling tools, and we know that within another 5 years, that number will have exceeded 1% of the population. So that's in the Western world, in particular the Far East and in the Indian subcontinent. We're in this acceleration in incidence phase where both incidence and prevalence are going up. And then if you look into the African continent in particular, you look at the very big populations, such as in Nigeria, for example, we're really seeing the emergence coming very rapidly now. And this has major global implications. If you look currently at the world's population, here, just sort of 2020 or so, and then you look where people are living by 2100, you can see that the European and North American populations actually potentially decrease a bit in size. The Asian continent gets a bit bigger and then starts to come back down to the levels where it's at now. But it's in Africa where this massive shift. These are young people growing older, and they're right at risk for development of a very significant IBD pandemic. So with the epidemiological trends, we can start to wonder whether or not IBD is actually an illness of modern living. And if you look across all the different data sets, the strongest epidemiological signal is urbanization. And it was in 2007, you had this tipping point where for the first time in human history, more people in the world were living in urban settings than those living in rural settings. So here we are, we've got a changing environment. We've got, as I said, an important role of the gut microbiota, and we have this genetic backdrop. And if you look together to try and make sense of this, we can think about the modern living aspect of the environment, and I'll come on to that. I've suggested already there's a reduced diversity or a dysbiosis in the microbiome, likely largely in response to these environmental drivers. And then if we look into our genes, we have a very fascinating story in terms of our response, our evolutionary response to pathogens, and that occurring relatively recently. The genetics of inflammatory bowel disease is where I started my research career, and over the last 20 years, we've uncovered many distinct areas of the genome, and now we can identify many distinct mutations in specific genes that are associated with the development of inflammatory bowel disease. One of those is the ERAP2 gene, and there's this very nice story from the last year where scientists in fact, Denmark and in the UK looked back at historical skeletons from both before and after the Black Death, this plague in the early 14th century that reduced population size, as you can see bottom right, by up to 50% in some areas. So very strong evolutionary pressure. And here you see that the ERAP2 gene, which is associated with the development of Crohn's disease, is actually protective here against bubonic plague and had a significant shift in its frequency, these variants associated with either Crohn's disease or plague. So here you can see the people that survived the plague because they had this variant in ERAP2 that gave them some resistance are now at an increased risk of developing Crohn's disease in the modern day and world. Similarly, the microbiome yields interesting insights, and I'm not going to talk too much about this today, but I will touch on the way that diet in particular has a very profound effect on our gut microbiome. The microbiome is fascinatingly complex, more genes and more cells than we have in our entire human body, as this figure illustrates. And again, just one snippet here. You can look relatively easy at archaeology sites where there are skeletal remains and you can extract DNA. You can actually now find fossilized poos that you can then discern to one specific individual and then do some sequencing of that microbiome genome. And they've done this here with these 8 red dots, 8 fossilized poos from about 2,000 years ago. And you can see that when they were compared to a group of both industrialized humans, that would be you or I in America or Scotland, and non-industrialized or modern, very rural, typically hunter-gatherer populations, you can see the shift. So our environment is shifting, but we also can see that our microbiome has shifted through these last 500, 1,000, and even our genomes have shifted in response to pathogens, as well as to modern life. And the environmental stimuli that are present in this modern world that we think are associated with IBD are diet, pollution, food additives, hygiene, antibiotics, surgery, stress, smoking, exercise, geography, travel, sunlight, and vitamin D. Some of these have strong layers of evidence, some they're a bit weaker. And I'll talk about evidence gathering in a minute. But let's turn our attention now to the dietary components that may, again that word may, have a causative role or at least a contribution to the development of IBD and thereafter also to the course of IBD over time. And you can see I've listed some here. We've got high-fat diet, emulsifiers, sugars, fibers, ultra-processed foods, polyunsaturated fatty acids, formula feeding versus breastfeeding, and propyzamide, this herbicide. And what I'm going to do is I'm going to go through each of these and tell one bit of each of the stories for each of these to try and give you a flavor of the kind of information that we're working with to answer this. But first, a couple of points. The Western diet is what we think of being harmful in many ways. You can take Western diet, you can call it the standard American diet, whatever you want. But this has been what we've been eating in the last 50 years. And you can see we've got a lot of dairy. We don't have particularly good fiber sources. We've got a lot of refined sugars and a lot of oils and high energy as well present. The sucrose as a proxy for at least part of the Western diet, the sugar, you can see here how things have shifted going from 1815 on the left-hand side of the x-axis to 1970. And we can think further about how foodstuffs have evolved from 1970 to the modern day. But you can see here this big shift in the last 200 years, in fact, which I think is quite telling. And here now you can look at oil consumption. The time scales changed a bit. Here we're going from 1909 up to 1990, and you can see these big shifts happening. And these are fairly comparable to the shifts that we start to see with inflammatory bowel disease. But we see with the Western diet, we see more meat, more animal protein, more refined sugars, more cooking oils, and less dietary fiber. And latterly, we see an increase, a very striking increase in the consumption of ultra-processed foods. Now, before we go into the details, it's worth just reflecting on a couple of points. The first here is that diet rapidly and reproducibly alters the gut microbiome. This is a very nice study from 10 years ago now, where healthy people were fed a normal sort of washout diet. Then they had a plant-based diet for a week. And then they had an animal-based diet for a week. And here you can see how the fiber intake goes up on the left. And then on the animal-based diet, it goes right down. Fat intake goes down, and then it goes right up. That's what you would expect. And then what you see is that when they went from this initial phase through to the animal-based diet, that within 2 days, the microbiome diversity plummets. And it's a reduced microbiome diversity and also a reductions in these essential fatty acids, acetate and butyrate, that shift and that are associated with the signatures that we see in inflammatory bowel disease. So with that, let's look at the evidence for the components of diet as causative in IBD. First, I just want to say a couple of things. Association is not causation. So if we say that diet A is associated with Crohn's disease, it might be causative, but it might not be. It might be that people with diet A are more likely to smoke. And we know that smoking has a causative contribution to the development of Crohn's disease, just for example. So replication of findings is key and is often lacking. Mice, where a lot of this data comes from, are not human, so we have to interpret mice data accordingly. Nonetheless, they do give us very strong evidence from well-conducted modeling scenarios. And diet is complex. Keep that in mind. We don't eat just sugar. We don't eat typically just one plant item. We eat food. And so it's our food patterns that are important, and diet is complex. And most of these studies try to reduce things down a bit. Human studies are full of bias and confounding. Just think of the person that you know that eats the healthiest diet you know. Do they also not smoke, drink very little amounts of alcohol, run or cycle regularly, have some kind of mindfulness practice? A lot of the people I know do. Similarly, you can have the stereotypical Scot who drinks iron brew, eats chips on a regular basis and pies, who's also significantly overweight, smoking, drinking a lot of alcohol, not moving around, and may also have a significant psychosocial detriment in their life. So these are complex areas that are difficult to untangle. And in many of the studies, we find that there is likely some residual confounding, even when the key things like smoking and BMI are taken into account. The other thing is that there is a lot of very low quality data published about IBD, and our press, conventional, and our social media networks are not always very discerning in terms of the quality of data, perhaps more concerned around the message that's given. And diet is a very good way to get a catchy headline. So do just bear that in mind when looking at the data. So with that in mind, misinformation, misreporting is rife by both mainstream media and on social channels. Okay, enough of that caveating. Let's look at the data that we have. This is a relatively new study from the Scottish Pediatric Group led by David Wilson. This was a very nice study done by Chris Burgess. And here what they looked at was breastfeeding in a population-based study. And for the most part, we have had this notion that actually breastfeeding is protective against developing IBD. Formula feeding is associated with an increased risk of developing IBD. But actually this, and also a couple of other population-based studies, suggest that that is not the case, and that in fact, breast versus formula feed has no impact on the subsequent development of inflammatory bowel disease. This merits a bit more searching. There's a very nice pan-Scandinavian study that we'll report quite soon. But overall, I think this shows us that we need careful studies. We need to test our assumptions. And we need to be very quick not to blame in any way an individual or an individual's mum or dad about the early life environment, because there may be some factors that are important with subsequent development of IBD, but there are others, like here, potentially breastfeeding versus not, that may have nothing to do with it at all. So we mustn't go anywhere close to any kind of blame culture. I think that's very important. One of the earliest papers that really drew me to the complexities of what we were going on here and made me really drawn to this gene environment, microbiota, and diet hypothesis is this. It's about 10 years ago. It's from Susanna Devkota, who's in Los Angeles. And they took this mouse experiment and they had, as you can see on the right, both wild type mice and genetically susceptible mice, here I think with an IL-10 knockout mutation. And what they showed was that you needed a high-fat diet plus this change in this host factor, this taurocolic acid, and this subsequent growth of Bilophila wadsworthia for, in the genetically susceptible host, an inflamed gut or colitis to develop. Really nice model of diet and host factor, microbial factor, and genetic background leading to the development of colitis. More recently, a lot of attention has gone looking at dietary emulsifiers. Dietary emulsifiers are the chemicals in foodstuffs that make food look and taste appealing. The classic example is up here. This is mayonnaise looking nice and yummy in a bowl with some parsley on top in the left, and on the right, without any emulsifiers present, doesn't look particularly appealing to me at least. And then Benoit Chassang has done a couple of very nice studies now, the first one of which published in Nature in 2015, demonstrating that dietary emulsifiers resulted in a reduced thickness of the mucus barrier in the gut, increased mucus barrier encroachment with bacteria, a decrease in the diversity of the microbiome, and an alteration in the microbiome composition, such that you got this translocation of bacteria across the barrier in patients who were given either carboxymethylcellulose or polysorbate 80. And these then went on to induce colitis at an increased rate compared to water as a control in doses that the FDA regards as safe and generally safe. So maybe 1 to 2% respectively concentrations of these two emulsifiers. And the data come more and more from Chassang's group, starting to look at the mechanisms by which emulsifiers may induce perturbed host microbiota interactions and subsequently triggering inflammatory pathways that lead to the initiation and or the perpetuation of the signal. Kevin Whelan's following this up in collaboration, in fact, with Benoit in the UK. Kevin's professor of nutrition down at King's. And they're doing this very nice study at the moment called ADAPT, where they're taking 150 patients with mildly active Crohn's disease. They then all go onto an emulsifier-free diet that's carefully mapped out for them. And then they have blinded supplements added back in containing about 25% of the food that they're eating over the 8 weeks. And that added food either has emulsifiers in or doesn't, but it looks the same, tastes the same, and is blinded to both the individual and the investigator, and then they're looking at outcomes. There's another similar study going on elsewhere, and in fact, there is a group in Leuven at the moment who are looking to see if they can determine which of the emulsifiers has the biggest effect in healthy people, working on the basis that perhaps it's difficult to remove all emulsifiers from your diet. And it really is. If you go to a food outlet of your choice, even if you want to go completely fresh, you're going to struggle to get some of your stock ingredients if you don't know where to look. It's complicated, in fact. And it might be that we need to look at finding the least dangerous emulsifiers that we can then ask the food industry to substitute for these ones that we think are causing problems. Sugars is next. Sugars is a really difficult thing to look at epidemiologically, because when people are unwell, particularly when people are unwell with gut issues, they tend to not eat very well. They're losing weight, and often people resort to sugar-rich foods and drinks in order to give them energy and help them feel better and get calories, etc., etc. Nonetheless, if we look at the animal models, and here's a paper published in Science Translational Medicine in 2021, you can see the impact of glucose in terms of worsening colitis in both DSS, this chemical-induced, and this IL-10 genetic-induced mouse model. You can see how different sugars had a different effect. Here you've got glucose, fructose, and sucrose. You can look at the incidence of colitis and in those different models there. And in fact, quite similar to the emulsifier story, there's a sort of trend here. So a degradation of the mucus layer and barrier function, dysbiotic microbiota, and a sugar-induced colitis that is still dependent on the microbiome. If you do this in a sterile environment, you don't get colitis. The big hot topic is ultra-processed foods, and ultra-processed foods have a lot of sugar, they have a lot of emulsifiers and lots of other things in them. And there've now been about 3 studies that have associated, to a certain degree, ultra-processed foods with the subsequent development of Crohn's disease, but not ulcerative colitis. And there are 2, I think, key studies here. This one's the PURE study. There's another one from the British Biobank, both of which are dietary cohorts that were looking to find dietary factors that associated risk of cardiometabolic disease and or cancer. And in the case of the Biobank, it was just a big study looking at outcomes in adults recruited between the ages of 40 and 65, mostly in the upper age band, primarily powered to look to see if you can find out why people are getting cardiometabolic, neurodegenerative disease, cancer, and other issues, not the development of Crohn's disease and ulcerative colitis. These are not kids. These are not teenagers. These are not young adults. These are all older people. And I think they have a slightly different phenotype. And they're looking to see if ultra-processed foods are associated with development of disease. In this paper, as I suggested, it does with Crohn's disease, not with ulcerative colitis. But importantly, when they fully adjust the model, it does seem to get closer going back towards the mean. And therefore, it looks like, there may be some residual confounding. So one needs to be very careful here. I think there may be a signal. We've known about Crohn's disease for a lot longer than we've been using ultra-processed foods. So this is not the sort of magic answer to why we're getting Crohn's disease, but it might be one answer. And nonetheless, if you look at what's good and healthy to eat, ultra-processed foods, it ain't, unless you are looking for a very rapid way to get calories in. And the current cost of living squeeze that we're particularly feeling is producing something of a perfect storm, really, because we have a lot of people that don't know very much about cooking and good, healthy foods. We have an unregulated food industry, and we have within that backdrop people who can't afford to eat healthily or at all, and ultra-processed foods offer a cheap way to get lots of calories into their family. The story about diets, similar in some ways, in that when you look at the diet data for fiber, and this is from the Nurses' Health study, a bit younger than the other cohorts, but still, this is not teenagers getting Crohn's disease and ulcerative colitis. And you can look and see that high levels of dietary fiber associated with decreased risks of developing Crohn's disease, no obvious signal in ulcerative colitis. And I think this is likely to be some kind of true finding. And certainly, again, for most aspects of diet, eating lots of plant-based fibers is a good thing to do. There are some interesting studies from across the world looking at this now, looking at different fiber intakes. If you look at traditional hunter-gatherer populations, they probably had a lot more diverse plant-based fibers than we do. And you can start to model this and look at the mechanisms by which fiber or Western diets fuel inflammation. In this study, they were comparing it to a group of hunter-gatherers in Tanzania in Africa. There are a couple of important caveats to this, actually. And this is the first one. So we've talked about fiber and we've talked about sugar, but let's just look at fiber first. This is a very nice study in man, in healthy people, in fact, but compared in a randomized manner the impact of high fiber diets and a diet that's high in fermented foods. And your fermented foods here are your kefirs, your kombuchas, your kimchis, mostly words that begin with K, apparently are good for you. But in this study, when they looked at the inflammatory effect and the microbiome, you saw actually a much better microbiome in a fermented food group than you did in the fiber-rich group. So that's something as a pause for thought. And then this other study in man looking at different sugars actually shows that artificial sweeteners, some of them have a profound effect on the gut microbiome. And when they did the mice studies here, they found that actually some of these artificial sweeteners have quite a big effect on the microbiome. It's not that clear whether or not it's positive or negative, but it does raise the suspicion that just cutting out sugars and going for artificial sweeteners instead may actually not be working in one's favor. There's a couple of other things I'm going to go through relatively quickly now because the mechanisms are quite complex, but there is an understanding about dietary polyunsaturated fatty acids now as a trigger of metabolic inflammation in the gut, probably in Crohn's disease, and that these may deteriorate the course of Crohn's disease by acting on this, this molecule called GPX4, but this does require targeted nutritional clinical data to validate. And then this fascinating story about this yeast, Deborah-Mikes hanseae, which appears to prevent intestinal wound healing, not cause intestinal wounds, but prevent healing. And the suggestion here is that if you have more of this yeast, that it changes the microbiome and then fails to allow, or rather propagates, the inflammatory process. So if you look at this Deborah-Mikes hanseae, you can find in high concentrations in inflamed versus non-inflamed. You can look at the wound bed length and look at the wound beds that are longer with lots of Deborah-Mikes hanseae in, and a variety of other different studies that have been done here that seem to support that. It's a very nice paper shown in Science last year. Where do you get these yeasts? Well, you get them in the rinds of cheeses and in the rinds of some cured meats and sausages and things. So it may be that consuming these yeasts in some of our prepared foods, some of our cured foods that, of course, we've been eating for a long time in the modern world, may increase the risk of Crohn's disease once other triggers have set it off in the first place. We then have one other fascinating story here, and this is where this herbicide propyzamide comes in. It's a complex narrative in this very intricate paper published in Nature last year, where they studied a wide variety of common environmental toxins. They then looked at those, screening them in a zebrafish model in the laboratory. They then used an artificial intelligence algorithm to then expand this out to see what else might be associated with these signals. And they honed in on this one chemical, propyzamide, which is a herbicide used to control grass and broadleaf weeds in a wide range and a wide variety of fruit and root crops. And this looks like it may be making colitis worse. So some of that's quite technical, but it's to show you that a lot of this data comes from both intricate mouse models, other experimental setups, with environmental data and epidemiological data that's present. There's not many of these stories where it all very neatly stacks up, and it's difficult to get to the point of causality with any of these things. Nonetheless, these do seem to be worth pausing for some thought on. So let's come back to a patient level. Let's go to a patient journey. You're born with your genetic environment, early life microbiome, triggering events leading to the development of IBD, remission, triggering events that lead to flare, and then the evolution of disease with complications, where in hopefully prolonged remission, at least some patients. So what's causing this first bit? What's causing IBD to develop? Well, we've got probably early life events that are critical: genetics, family history, the microbiome, etc. Weaning, maybe. We've shown some contrary data to that in terms of breastfeeding. Early life environment, lifestyle, hygiene, and antibiotic exposure does, though, seem to be important. And so we need to study people before they've developed IBD to look at this. And ideally, not just before they've developed IBD, but before the inflammatory process starts to proceed. What about dietary therapy to treat IBD, both to induce and maintain remission? Well, this is complex, and I'm not going to dwell too much on this today because it's a whole nother talk. Suffice to say, exclusive enteral nutrition works. It works in kids. We know that. It works as well as steroids in kids. We know that. I know that it works in adults because we use it all the time in our practice instead of steroids, particularly in small bowel Crohn's disease. Makes people feel better, replenishes the nutritional defect, works as well as steroids, improves biomarkers, induces mucosal healing, and can be drunk via drink, via straw, or via a nasogastric tube. A group across the UK are about to look at enteral feeding prior to surgery in adult Crohn's disease. This is a big pragmatic study with over 600 patients, so we look forward to the outcome of that. Two other diets that are talked about a lot are the specific carbohydrate diet and the Mediterranean diet. This study from Jim Lewis, published 2 years ago now, randomized patients with Crohn's disease to either Mediterranean diet or the specific carbohydrate diet and looked to see what the outcomes were. There was no control arm with neither diet. And what they showed here was that both of the studies helped people get better. Neither was superior to the other. This study shows that complex diet studies can be done. It's reasonably well powered, but there is room for improvement in the study design to really try and work out what works and how to use it. My advice to patients, cutting to the chase, is to follow something close to a Mediterranean diet because we have a lot of good evidence around that for many health outcomes. And in fact, a specific carbohydrate diet is a slightly IBD specific thing that doesn't have quite such ease. It's quite restrictive. And I think it lacks really good data to support its use in IBD. The anti-inflammatory diet is another one. This paper from India was published in Gut a year ago. And in this study, it's an interesting idea. The study has flaws in it, but it's an interesting concept. Take patients with active ulcerative colitis, give them a fairly intensive fecal microbial transplant or a stool transplant regime over 8 weeks. And then after that, randomize them to either just be on normal therapy or to have a diet plus normal therapy. And they looked at some of the clinical outcomes here and showed that people that followed the diet afterwards did a bit better. What about diet to maintain remission? This is what we're looking at in our PREDICT study, trying to work out what diet factors are associated with disease flare. This is a big study powered to look at diet, environment, genetics, and the microbiome, which factors are associated with and predict disease flare, trying to answer questions about what patients should eat, how they should stay well, and what's the impact of IBD on mood, lifestyle, exercise, and sleep. So here's the study design: 2,500 people with IBD in clinical remission, with a detailed dive into the microbiome, into a variety of questionnaires and diet, and then followed over 2 to 4 years to then look at those that flare versus those that do not. Now, the primary data for this study has just been accepted as a late-breaking abstract in the clinical plenary at DDW. So I will be sharing those data for the first time in just over a week, and we'll be posting a YouTube video for those that can't attend at the same time with the results. That's a week on Sunday. And that's taking the food frequency data that we have from just over 1,000 of the participants. Note, this is an all-ages study. We've got 47 children under the age of 16 at study entry, which we think is a very important contribution to the study design. Interestingly, we asked already patients what they thought caused their diseases to flare. Stress was number 1. Dietary changes was number 2. We also asked people what diets they used to control their symptoms. We found that many people did use some kind of diet. In Crohn's patients, more likely to follow low-fiber diet, as you'd expect. Quite a lot following gluten-free and low-lactose studies, and about 10% who'd had exclusive enteral nutrition for their Crohn's disease. So the last part of my talk is thinking about what we can do practically today. Advice to patients, parents, pressure on policymakers, research, and the challenges and the opportunities. This is a summary from the IOIBD consensus group looking at lifestyle behavior and environmental modification for the management of people who already have IBD. And you can see some obvious things here: avoid smoking cigarettes, but you also see adopt diets that have proven best in clinical trials. And I've shown you already, we don't have a lot of really good clinical trials with diets apart from the enteral nutrition. And then lifestyle, behavior, and environmental modification, advice to decrease the risk of IBD in children. Encourage breastfeeding where possible, although I wouldn't be too prescriptive about this, but we do know that breastfeeding has many advantages. Avoid unnecessary antibiotics in early life. Avoid secondhand cigarette exposure. The GEM study suggesting having a dog as a pet is helpful. Limit consumption of processed and ultra-processed foods, and plenty of plant-based fiber. These ones at the bottom, I added in. They're not in that consensus statement, but I think they are probably sensible things that will improve many health outcomes, not just IBD. Because diet is probably the main modifiable environmental risk factor that we have, apart from smoking. And there are many real opportunities, not least in terms of better health, better energy, reducing type 2 diabetes and obesity, but also I really believe in terms of improving IBD outcomes as well as mitigating cancer risks. But there are major challenges. They're behavioral change and adherence, key skills and knowledge, like simple things like what ingredients are and how to cook, and the cost of living crisis. So all of this requires a solid evidence base, not just for the diet, but also at a social level in terms of how do you implement behavioral change in a society. And meanwhile, I do urge us all to approach the topic with sensitivity. Diet does not per se cause Crohn's disease or ulcerative colitis. Many people develop Crohn's disease or ulcerative colitis whilst eating a very, very healthy diet their whole life. Adherence is a very important watchword with diet. We know this from weight loss studies. People can maintain a diet for a short period of time when they can see a visceral result, but it's only short term for many people. Food's essential for life, so you can't avoid it completely. Processed foods are designed to be highly addictive, and we must try and tackle that, I think. Diets are hard to maintain. In IBD, will a dietary strategy be required long-term, like a drug maintenance, or not? And I think we need long-term, pragmatic, real-world studies to try and address this. But I do believe that dietary therapy, maybe on its own for some people, but for many people as an adjunct to medical therapies, which are getting better and better, will enable us to break through the therapeutic ceiling that we currently see sitting at about 30 to 40% of outcomes in our patients. So to summarize, I don't believe that diet is the cause of IBD, but I do believe it contributes strongly to etiology, and maybe it's doing this largely by being a driver rather than an initiator of inflammation. So I think dietary therapy for IBD will be an important adjunct to medical therapy, and perhaps work really well if we can have it as an adjunct to medical and microbial therapies. Prevention strategies based around diet seem attractive, but we need more data. We might be able to prevent disease flare and disease onset if we get this right. Compliance with dietary therapy is a major personal and societal challenge. So please do treat the whole of this conversation with sensitivity in the clinic and in the home. Thank you for your attention. I look forward to some discussion.
All right. Thank you, Dr. Lees. We really appreciate that. I know the chat box has been going with some comments. Jean Tung, a physician from Oklahoma, mentioned, thank you for mentioning food disparities and cooking abilities or lack thereof. So really appreciative of that comment. And then there was also a question from Dr. Hillary Michael. With the efficacy of EEN for induction, what are you using for maintenance therapy? This is often our struggle.
So there are some people who don't need maintenance therapy for Crohn's disease. I think that's different in an adult population than in a childhood onset population, but nonetheless, it does occur at all ages. So there are some people in whom induction with exclusive enteral nutrition is sufficient, but most of the time it's not. And in that situation, well, it depends what you're going to use for maintenance therapy. Depends very much then on the clinical phenotype and the risks. For us, a lot of the time, that would be an anti-TNF such as adalimumab used as monotherapy. That might be a very good option for many people with a moderate risk phenotype with Crohn's disease. But I might, in some people, just follow the biomarkers. If the patient has improved clinically, if the CRP has normalized, if the calprotectin has normalized, then I will follow those, maybe on no therapy. And if I follow those every 2 months and they remain low, then I might just hold my horses with any therapy.
Okay, great. Also another comment, thank you for your research. We look forward to the preliminary data. Those that can't make it to DDW are appreciative of the fact you will be sharing it in other channels as well. Another question from, it says, does Dr. Lees think that an approach would be to develop a food product that is IBD healthy, such as a shake or other product?
Yeah. It's an interesting question. So I had a very interesting guy, Joe Sabaro from Leuven, and his approach to this is that we have failed for decades to regulate the food industry. And they've made all these many billions in profit from developing processed and ultra-processed foods that store, easy, convenient, addictive. Problem is that many of them are not very healthy. But if we can work out some of the worst components of them, and emulsifier is an interesting thing, actually, because mayonnaise in itself is not an unhealthy thing to eat. But if you could have a good emulsifier in there to your mayonnaise, then that would be helpful. So that's one component. In terms of things that are healthy for, food products that are good for IBD patients, like a shake, well, that's basically what your polymeric drinks are, in a way. So that's one option. How much of what we do moving forwards is going to be removing things versus adding good things in. And behavioral change, as I've mentioned many times, by removing things is challenging. So it's attractive to have something that might add in things that are beneficial. I think it's likely to be a combination. Remove things that we learn more and more are harmful, add things in that are going to be beneficial, both in terms of controlling the inflammation, like our anti-inflammation medicines that we rely on, but also dietary components that may help to replace. In many ways, this is what we do anyway with diet. We look at probiotic or prebiotics with dietary fibers and fermented foods that then improve the microbiome that will likely then improve IBD.
There's another question that came in. In the scenario where you start anti-TNF for maintenance, why not simply start with the anti-TNF?
Sometimes we do. I think there is some evidence in pediatric literature that if you start with both, you get a better outcome. Sometimes it's not practical to start with anti-TNF within a day, a week, a couple of weeks, or even a month. Depends on the situation. And I think this will depend on where people are at and what's going on. In my clinical practice, what I do, and I appreciate this may be different in other areas, is that if I see a patient with what I think is going to be a new diagnosis of Crohn's disease, small bowel Crohn's disease, then I may well start enteral nutrition straight away. I do that by mouth with polymeric shake. That's for the person that comes with weight loss, the person that comes with significant abdominal pain and robust inflammatory markers. But I need to get their blood workup done. I need to do their biologic screening. I need to scope them. I need to do their scans and all the rest of it. And with the best will in the world, it's going to be 2 to 4 weeks to have all that available. The enteral nutrition in that time period, rather than the steroids, will get the person feeling better because we see the pain reduce, because you then have no real fibers within the gut that's often account for the pain, and you start to correct the nutritional imbalance.
One last question, Dr. Lees. Any data on post-surgery patients for medication versus diet?
Not that I am aware of that is very robust. It's an interesting avenue. The Crohn's recurrence model is also a good one to study, because in some ways it's close to the situation prior to someone getting Crohn's disease in the first place. Although I do worry a bit that is overstated. It's still a somewhat artificial model, but nonetheless, it's the closest that we get probably to that in people who already have Crohn's disease. Most important thing after surgery is to not smoke. I think that for many people, either a very close monitoring and or a combination of antibiotics and therapy, be it an anti-TNF or be it vedolizumab, as per the data that was presented at ECHO this year, is beneficial. But I would also think about diet. But in that situation, I would tend to be relatively non-prescriptive. After surgery, people will have a lot of adjustments to make. We don't have any particularly good evidence that there is dietary changes to be made there. I would be telling people, right, good time to think about being healthy. I would eat something close to a Mediterranean style diet if you can. But bear in mind, people will have to adjust what they eat a little bit in at least the first couple of months whilst they get used to having had resectional surgery and the alterations that then has on their gut function. Many will have diarrhea for a period of time. You don't want to change things around too much. And some may have actually become quite nutritionally deplete prior to the surgery, and actually the priority may just be to get them back to a good nutritional balance. So I'm quite pragmatic with it and not prescriptive, despite my interest in this, and that's until we have, I think, robust data come out and understand better and are better resourced to deal with all of this.
Thank you.
All right. Well, we do have some questions that were listed in the chat box that we are not going to be able to get to today in the interest of staying on time. So we do plan to follow up with those and send those to you, Dr. Lees. We appreciate you coming. Any final words, Brendan, Sabina, or Dr. Lees?
Thank you very much for joining us.
My pleasure. Thanks, everyone.
Fantastic.
Okay. Thank you.